Hello students, welcome to the class of teacher Meenal Pereira. In this video, we will begin with lesson number 8, that is cell biology and biotechnology. The points that we will be studying in this lesson are, we will be studying about cell biology, which is also called as cytology, then about stem cells, biotechnology and its applications, and important stages in the development of agriculture. So let's begin. So first we begin with what is known as cytology. Now children, you already know that cytology means study of cells. This we have already studied in the previous year. So it includes the cell structure, its types and various organelles. Beside this, it also includes the study of cell division and many other aspects of the cell. We have studied, children, two types of cell division, meiosis and mitosis. So that is also included in the study of cell. There is revolutionary change in the field of human health due to cell biology. There are various research centers which are established at Pune and Bangalore in India that studies about the cell and how the cells could be used for the development of human health. Okay, so you can visit the site given in this video and you can find more information about it. Okay, now next we begin to what is called as stem cell. Now what are the stem cell? Basically, these are the special type of cells which are present in the body of multicellular organism. And these cell gives rise to all other types of cell present in the body. So this cell also plays an important role in wound healing. Okay. Now we have studied in the previous standard that stem cell of plants we have already studied. Now we will be studying about the stem cells of animals, particularly human being. New organism is formed from the zygote, that is formed by union of male and female gamete. Now this diagram shows you the formation of zygote, which is a result of sexual reproduction where the fusion of gametes takes place. At the earliest stage of the development, the organism is in the form of mass of cell. Okay, so here you can see just a mass of a cell. It is not differentiated. At the time, the cell or the what you can say is the cells of the mass are all alike or same. So these are called as stem cells. Okay, so you can see in the diagram, you can see the mass of cell, that is a ball of cell. And later on, this cells or from this cells we could get different type of cells. So these cells which are at the early stage or initial stage which are almost alike or same are called as stem cells. Now how can we do the stem cell preservation? For the preservation, stem cell samples are carefully collected from the source like cord blood. So this is the umbilical cord by which the child is attached to mother's body. Okay, so it is removed from the cord blood or red bone marrow or embryo, which is also the blastocyst, and it is kept in small sterile vials. So these are the special vials which are small and sterile, that is no microorganisms are present into it, and then those are stored in liquid nitrogen at about 1350 to 1900 degrees Celsius. Okay, so that is minus, so that it remains in a good condition. Now stem cell research. In biotechnology, stem cell research is a revolutionary after cloning. This technique has the potential of bringing about the fundamental changes in the medical science. Now, depending upon the source, there are two types of cells. Embryonic stem cells, that is in the embryonic stage, that is the first stage, and adult stem cells that are removed from the adult body. Let's see more about it. Coming to embryonic stem cell. Now, as I already told you, division of the zygote starts and thereby it is converted into embryo. Okay, so This is in the diagram, we can see the embryo, which is again a mass of cell. Cells of embryo undergoes repeated mitotic division. So by mitosis, the number of the cells are increasing. The cell differentiation starts from the 14th day of conception, that is after fusion. Cells of different organs like osteocytes, that is bone cells, and hepatocytes, that is liver cells, and neurons are formed due to differentiation. Embryonic cells before differentiation are called as embryonic stem cells. So at this stage where the cells are not differentiated, so these are called as embryonic stem cell. So as you can see, this small, these are embryonic stem cells, and later on they can be differentiated into various cells. 220 different types of cells in human body are from a single type of cell, that is embryonic stem cell. So these are the primary types of undifferentiated cells with self-multiplying ability, and they are the parent cells of all the human types or human cells. Okay, so this embryonic cells, they undergoes mitosis, they are multiplying themselves, and thus they form the maximum cells in the human body. And this property of stem cells is called as pluripotency. Now what is observed that if these stem cells are collected before the differentiation, that is between the 5th to 7th day, and cultured with certain biochemical stimulus in the laboratory, then they can transfer themselves into desired type of cells, thereby tissue, and finally into organ. Okay, so what I'll do is I'll remove some embryonic stem cells, and instead of forming the entire body, now suppose I want liver. So what I'll do is I will give some biochemical stimulus, and I can develop the cells of liver. Then the cells will form tissue, and the tissue will combiningly form together the organ liver. Okay, isn't it very revolutionary, children? Then we have the adult stem cells, which are obtained from the adult body. As I told you that it is obtained from bone marrow. Bone marrow is a red color substance that is present between the cavities of the bone. Then it can be also obtained from the adipose connective tissue and blood. And as we have already studied in the previously, it is also obtained from the blood cord immediately after birth. And very similarly, these cells could be differentiated into different type of organs. So two type of stem cell, embryonic and adult stem cell. Now what are the uses? Now the first use is the regenerative therapy, that is a cell therapy. Stem cells are used to replace the dead cells in the conditions like diabetes, then the myocardial infarction. Myocardial is heart diseases, the Alzheimer's disease, which is a disease where a person tends to forget the thing due to aging, and Parkinson's disease, which is a mental disease that leads to instability. That means the person's movements are not proper. Okay, so these problematic stem cells or these problematic cells could be replaced. To reduce blood cells required in the conditions like anemia, thalassemia. Thalassemia is a condition where children, the hemoglobin production of body is very less. So that leads to severe anemia, and that could even leads to death. And also leukemia, that is cancer. So all these type of cells, problematic cells, could be replaced by the regenerative therapy. Then organ transplantation. In case of failure of the organs like kidney or liver, and this can be produced with the help of stem cell and then transplanted. So as we have discussed in the previous slide, before the differentiation, the stem cells are reduced with the biochemical stimulus. It could be, now this is a cardiac cell, that is a cell of the heart, muscle cell for the muscles, and intestinal cell for intestine. So whichever organ I want, I could develop that. Of course, children, it is not so simple as we are talking, but it is a very revolutionary concept. We can actually do it with the help of biotechnology and stem cells. Now, organ transplantation is a very, very important concept. We all know that the various organs in the human body either become less efficient or completely functionless due to various reasons like aging, accident, infections, disorder. Life of such person becomes difficult or even fatality may occur under such condition. Fatality means death. However, if the person gets the necessary organ under such condition, its life can be saved. Availability of donor. Donor means the one who wants to give the organ. So this is a very important factor. Each person has a pair of kidney. We all know that. As the process of excretion can occur with the help of single kidney, person can donate another one. Okay, so if one person wants to give its one kidney, can be given, because on one kidney the function person can live a very normal life. Similarly, skin from certain parts of the body can be donated. Okay, so we can donate our kidney because we have two, or skin. Okay, so that can be donated. Now here children, you have to remember that these are the organs that we can donate when we are alive. Okay, but there are certain organs which we cannot donate until and unless we are dead. Like I cannot give my heart or I cannot give my brain until and unless I am alive. Okay, so alive, what I can give is I can give liver, I can give skin, or maybe blood, which is also a very good thing. Okay, so various factors like blood group, disease, disorder, age of the donor and the recipient needs to be paid attention during transplantation. So this is a major surgery. So all the factors needs to be taken care. Okay, but there are certain organs like heart and lungs, as I said, then intestine. So these are the things which cannot be donated, liver, when a person is alive. Okay, so it can be given only after death. So this leads to the concept of that is after death. That is the posthumous donation of body and organs. And it is really good, children, because after death, the body parts are really, like you can say that after the death of the person, the body parts are of no use. Okay, so then what we are doing is traditionally we are burning the body or we are burying the body. But then the valuable organs of the body are getting wasted. So if we give it to someone else, okay, a person can very well live a good life with that. Okay, so therefore concept like organ donation and body donations have recently become very popular. Okay, because these organs could be saved and can be given to needy person. A liberal view behind the concept of organ and body donation is that after death our body is of no use to us. Awareness about these concepts is increasing in our country and people are voluntarily donating their bodies. Okay, just imagine, children, a person who is blind, who cannot see from its childhood. Okay, and after your death, your eyes are of no use to your body. So if these eyes are donated, the person can see the life. Okay, so you are not dead, you are not decomposed by the microorganisms, but in form of your organs, you are live everywhere or in the person's mind or in the person's body. So blinds can regain the vision. Lot of people can get organs like liver, kidney, heart valve, skin. Okay. Similarly, body can be made available for research in the medical studies. Okay, nowadays you all know that coronavirus. So this scientific study could be done on the body. Many government and social organizations are working towards increasing the awareness about body donation. There are various stars, various celebrities who are also doing this awareness campaign. Okay, and very often we read in the newspaper that those persons who are dying due to any accidental death, their organs could be donated. Okay, so be a thinker, be a donor. Okay, so see, this one person can one person's organs could be given to 9 persons. So you could change 9 lives. Okay, so don't simply waste your body after death. Okay, it could be used for some person. So it is a very good concept and everyone should think about it. Okay, so this is a new concept. Okay, so I hope you all understood this. So in this video we studied about what is called as cytology or the study of the cell. We studied about the stem cell, okay, and we also studied about the organ transplantation and donation. Okay, so I hope it is clear to everyone. Thank you very much. God bless you.